Okay, Hardy-Weinberg population genetics. There are not a lot of equations to cover in biochem, but we need to use some here. These equations are used to describe the frequencies with which alleles of a gene occur in a population. Here we'll use P and Q to refer to how common two different alleles of a gene are. So if we're talking about the alleles big A and little a, this would mean you have a P% chance to have a big A and a Q% chance to have a little a. So what does Hardy-Weinberg equilibrium mean? Basically just that the ratio of P to Q will not change over time if certain conditions are met. If two alleles of a gene were not in Hardy-Weinberg equilibrium, why might their relative frequency change over time? The main reasons are if one gene is mutated, or if one gene gives a survival advantage over the other one, or if mating in the population depends on having one gene instead of the other one, or if individuals leave or enter the population. Like if a bunch of individuals who are homozygous for little a enter the population, then P will decrease and Q will increase. This means that if you want to keep the ratio of P and Q the same, which is Hardy-Weinberg equilibrium, four things have to be true. No new mutations can occur, neither big A nor little a can give a selective advantage. Mating has to be random and not based on the genotype, and there can be no migration into or out of the population. If all of these conditions are met, then the ratio of P and Q will not change over time, or in other words, they are in Hardy-Weinberg equilibrium. So why is this important? It's because it allows you to do some simple calculations to learn about the prevalence of individuals with a disease or carriers of a disease if you know the allele frequencies. We'll start with the basics. P plus Q equals 1, since we're assuming that P and Q are the only two alleles of this gene in the population. If 40% of the alleles are P, then 60% are Q, so the total is always 100%, or 1. If P equals 10%, then what's Q? Right, Q would be 90%. Okay, that one was too easy for you. Let's take it to the next level. So how about this other equation, P squared plus 2PQ plus Q squared equals 1. Let's break it down. P squared and Q squared are the proportions of the population that are homozygous for the P and Q alleles respectively. So if we use the same example as above, if a gene has a 10% chance to have the big A allele, then P is 0.1. So the probability of being homozygous for this allele is going to be P squared, or P squared equals 0.01, so you have a 1% chance to be homozygous for big A. And similarly, since Q is 90%, or 0.9, then what proportion of individuals are homozygous for the Q allele? Right, so that would be Q squared, or 0.81. Now 2PQ is the proportion of heterozygotes. Since individuals can either be homozygous for P, homozygous for Q, or heterozygous and have one of each allele, the sum of these three have to equal 1. And why is it 2PQ instead of just PQ? That's because you can either have big A little a or little a big A. So there are two different ways you can be heterozygous. Okay, so let's do an example of something you might see in an exam. Let's say 4% of individuals in a population have an autosomal recessive disease. And you need to figure out what percentage of the alleles in this population are healthy. This means that 4% of individuals are homozygous for the diseased allele. We can assign P to be the frequency of mutant alleles and Q to be the frequency of healthy alleles. So what does P squared equal? Right, this would be 0.04, or 4%, since P squared is the proportion of individuals that are homozygous for P, and that was given in the question as 4%. You can solve this for P by taking the square root of each side, which gives us P equals 0.2. Now the question is asking for the frequency of healthy alleles, which is Q. So we just need to solve for Q and we'll be done. We can plug our P equals 0.2 into our equation P plus Q equals 1 to figure out that Q equals 0.8. So 80% of alleles are healthy. And that's it. Okay, let's do a flash quiz about genetic terms. So what is the term for a gene that, in a mutated form, can cause a variety of different phenotypes? This is an example of pleiotropy, and I also want to point out here that many people might study these by looking at the term and trying to remember what it means. But you should also spend time doing the opposite, which is look at the definition and think about what term it corresponds to, because the way you'd see this on the exam is that a disease would be described, and they'll ask you to describe what genetic term this is. Okay, as promised, let's return to imprinting. Do you remember what that means? It's when a gene's expression, and therefore the phenotype it produces, depends on which parent it was inherited from. For example, let's say in one person, the allele from one parent is imprinted, which is the black one here, and the other is expressed, which is the orange one. Imprinting can be established by methylation of the gene's promoter in one parent, so it won't be expressed. When this person produces gametes, in the germline, these old imprints are erased, leaving a blank state. Then, depending on the sex of the individual, new imprints are made. So in this case, the gene can be expressed in chromosomes inherited from the mother, but not in chromosomes inherited from the father. Now let's say this gene has a mutation in it right here. The mother will not be affected, since this copy is imprinted anyway, and she has another copy which is expressed. However, if her offspring ends up with that copy, then they're in trouble, since the only other copy they get is from the imprinted version from the father. This is the case in Angelman syndrome, in which the maternal allele is deleted and the father's allele is imprinted. Patients with Angelman's will have intellectual disability, seizures, ataxia, and inappropriate laughter, and the combination of these has led to its nickname of the happy puppet. Now, Prader-Willi syndrome is similar, but is due to a deletion or mutation of a normally active paternal allele and an imprinted maternal allele. So it's basically the exact opposite as Angelman syndrome. Patients with Prader-Willi will have intellectual disability, hyperphagia, obesity, hypogonadism, and hypotonia. The genes that cause both of these are in chromosome 15. These syndromes can also be caused by uniparental disomy, as I mentioned earlier, because if a gene is imprinted by one parent and the offspring receives both alleles from that parent, then they won't have a copy of the gene that they can express. There are a few different ways that genetic traits can be inherited. Remember, what we're talking about here is inheritance of phenotypes or traits, not inheritance of genes. As I talked about before, a gene can come in different versions called alleles of that gene. For example, for gene A here, a certain type of mutation, like a nonsense mutation, can cause the gene to not function at all. Remember what a nonsense mutation is? This could be a premature stop codon, so that allele would probably have no function, and I'll designate that as little a. Now, if the allele of that gene on the homologous chromosome is normal, like big A here, you probably would have no phenotype, since one functional copy is usually sufficient to do the job of that gene. So in this case, the trait or phenotype that we're interested in is what you see when both copies of the gene are abnormal or deficient, and the lack of the trait means that at least one copy is functioning properly. So what type of inheritance pattern is this? This would be autosomal recessive. To inherit an autosomal recessive trait, both parents have to be at least carriers, which means they have one functional allele and one defective allele. So they don't have the trait, but they can pass it to their offspring. If they are both heterozygous, then what's the chance that their offspring gets the defective allele from each parent? They would have a 25% chance, because each parent has a 50% chance to give a defective allele and a 50% chance to give the functional one. So here I've drawn a Punnett square where the column headers represent one parent and the row headers represent the other parent, and there's 4 possible choices for the offspring. you can see two of these are going to be heterozygous, and one will be homozygous recessive, which will be affected by the disease. So this means there's a 25% chance that this person is affected. So what's the probability that the offspring of two heterozygous parents is also a carrier? That would be 50% because there are two possibilities out of the four total possibilities. Alright, let's move on to gene B. So in gene B, we're going to have a different type of mutation. This will be a frameshift mutation. Do you remember what that is? A frameshift mutation is when you insert or delete nucleotides in such a way that it throws off the reading frame of codon triplets. So all of the amino acids downstream from the affected codon will be incorrect. Since we have a whole new amino acid sequence, this mutation might create an allele of the gene with a new function. And we'll call this one big B. So in this case, as long as at least one chromosome has this mutant allele of the gene on it, that new function will be manifested in this cell. What type of inheritance pattern is this? This will be autosomal dominant. So how are autosomal dominant traits inherited? You only need one copy, so only one parent needs to have this allele. But that means that that parent also has to have the trait. Okay, what about sex chromosomes? Let's now pretend all the big chromosomes are X chromosomes and the small one is Y. So on the left, that means we have a female, and on the right, this would be a male. Is an X-linked recessive trait more likely to show up in a male or a female? X-linked recessive disorders are more likely to occur in males because males only have one X chromosome. So if the gene in that chromosome is affected, then they have the trait. Which parent will males inherit an X-linked recessive disease from? They're going to get this from mom because they get a Y chromosome from their father. Females need to get two affected copies to fully have the disease, which is very uncommon. Now why do I say fully have the disease? Well, as I mentioned earlier, female cells inactivate one copy of the X chromosome. So only cells that have the X chromosomes that carry the disease as their active X will be affected. However, this will be a less severe phenotype than in males, and in many cases is not even detectable. For example, color blindness is due to an X-linked recessive mutation, but if even half of the female cells are able to detect color, she will probably never notice that anything's unusual about her vision. Now, if a female gets a defective allele from each parent, then what do you expect to see in her parents? Well, her mom could be a carrier, so you wouldn't expect any phenotype, but her dad must also have the trait, since dad only has one X chromosome to give. So if he gives his daughter a defective allele, then he must also have that trait. Now how about inheritance of X-linked dominant disorders? If you were looking at someone's family tree, what would be the giveaway that the familial trait is X-linked dominant? Well, these can be inherited from the mother or the father. But if a man has an X-linked dominant trait, he will give it to all of his daughters, since he only has one X chromosome to give them. The last inheritance pattern we'll cover is mitochondrial. Since everyone's mitochondria come from their mother, these can only be passed down through the maternal lineage. Also, all of the offspring of an affected woman will be affected. Examples of these diseases include mitochondrial encephalopathy, myoclonic epilepsy, and Leber hereditary optic neuropathy, which causes degeneration of retinal ganglion cells and axons, ultimately causing loss of central vision. So why might these particular cells be affected? Well, mitochondrial defects will cause the most problems in cells that require lots of energy, which includes retinal ganglion cells and particularly active neurons. You may also see ragged red fibers on muscle histology, which are clumps of diseased mitochondria. Alright, so how are you going to see these inheritance patterns on your exam? Well, one way you can be tested on inheritance patterns is that you might be asked what the probability is that someone gets a disease after being given some other information, which might include a family tree. An easy way to do these is to draw a 2x2 grid called a Punnett square, kind of like what I drew earlier. This allows you to see all the possible combinations of genotypes for an offspring of two parents. So let's say two people who are carriers for an autosomal recessive trait but don't have the trait themselves have a child. What are the odds the child will have the trait? So once again, this will be 25% because only one of the four possibilities is affected, is heterozygous recessive. But now let's do a different kind of example, this time with sex chromosomes. So let's say you have an X-linked dominant trait, and we'll draw the father up here, and the mother will be in the rows. So again, the father is going to be XY. So if the father has the trait and it's autosomal dominant, and the mother does not have a trait and is not a carrier, and we use the same nomenclature as before, so big B is the affected allele and little b is the normal one. So now the child can only get the X little b from mom and either X big B or Y from dad. So what proportion of daughters do you expect to be affected by this trait? Only the offspring with two X's are females, so all of them will be affected. What about sons? 0% will be affected because they all have X little b and Y. So one more way you might be tested on this is to have to figure out what the inheritance pattern is by looking at a family tree. So let's do some examples, and don't cheat by looking in the book. The best way to do this is by process of elimination, using the things we talked about earlier. So I've listed all of them here as a summary, and you should definitely take the time to memorize these, although if you understand the concepts I just went over, you can actually figure them out easily by thinking about them instead of by memorizing anything. So in summary, for autosomal recessive diseases, you'll usually see them in only one generation, since carriers don't show the trait. And what I didn't tell you before is that these are a lot more common in cases of incest, since members of the same family can both be carriers. Autosomal dominant diseases are generally in all generations, since one person can't have it unless one of their parents has it also. X-linked recessive diseases can only be passed to a male from his mother, since he gets the Y chromosome from his dad, and if a female is affected, then her dad must be affected too, since she has two affected copies and her dad only has one X chromosome to give her. For X-linked dominant, you should know that an affected male will always give it to all of his daughters, but won't give it to any of his sons, since they get his Y chromosome, and mitochondrial traits can be passed from an affected woman to all of her offspring. And by the way, they might give you a key on the exam, but remember that males are always represented by squares and females are circles. And if it's black and white, then the affected will be black and normal is white. In this case, affected is shown by orange and normal is purple. Alright, so here's the first example we'll go through, and if you want to pause the video and think about it, go ahead. So like I said before, let's use process of elimination. We can see that this is in all generations, so it's not autosomal recessive. We have an affected female without an affected father, so that means it's not X-linked recessive either. And we also have affected women who don't give it to all of their offspring, so we know it's not mitochondrial. Now this one becomes a bit tricky because technically it could be either autosomal or X-linked dominant, but we have to pick the most likely one. The clue here is that the grandfather gave it to both of his daughters, but not his son, which means there's a really good chance that this is X-linked dominant, since an affected male only has one X chromosome, which is affected, and he passes it to all of his daughters but not to his sons. So the answer to this would be X-linked dominant. Alright, what about this one? So here we can see again that it's in all generations, so it's not autosomal recessive. An affected mother isn't passing it to all of her offspring, so therefore it's not mitochondrial inherited. And an affected male is getting it from his dad, which means it's not X-linked dominant. Lastly, we have affected females whose fathers are not affected, like in this case, which means it can't be X-linked recessive. And that leaves us with just one answer choice, which is autosomal dominant. Alright, I'll do just one more. So in this case, only one generation is affected. And as far as the step 1 is concerned, this will pretty much always mean that it's autosomal recessive.